Hi, I'm Kalpana Hanthanan Arachchilage. I'm a postdoctoral scholar in Dr. Wang's research group at University of Wisconsin-Madison. Today I'm going to talk about our efforts to infer disease-specific gene causal network using pseudotemporally ordered gene expression data. As you can see in this figure, if you have control and disease cell trajectories, sometimes you can identify distinct variations in their developmental paths. In this project, we are trying to identify these disease-specific trajectories and use them to infer gene causal networks. So this is the computational pipeline that we are using. First, we start with single cell expression data, or in this case, scRNA-seq data for disease and control samples to initiate our pipeline. Then we use nonlinear dimensional reduction and cell clustering to group these control and disease cells to specific clusters. Then we infer trajectories for control and disease samples separately and align them on top of each other to identify control-specific, disease-specific, and conserved developmental paths. By doing that, we can obtain pseudotemporally ordered gene expression for different genes. By providing these pseudotemporally ordered gene expressions as an input to our causal inference approach, we can identify causal links between different genes or transcription factors and target genes. Then finally, we can compare it with gene regulatory networks. We can identify GRN-supported causal links. Okay, so the causal inference technique that we are using is convergent cross-mapping. It's an approach developed by Sugihara in 2012. So first I'm going to explain the basic working principle of CCM. Let's say we have two time series variables, x and y. And if x causes y, according to CCM, we should be able to predict variable x using the values of variable y. First, we use delay embedding to obtain shadow manifolds for both x and y. Since we are trying to see whether we could predict x using y, we then estimate a new manifold using the values of y manifold, or the points of coordinates of y manifold. So we are trying to see whether how similar the predicted x manifold and the original x manifold, and see whether how similar they are. If they are similar, that means we can predict time series x using time series y, or in other words, we can say that x causes y. Okay, so and the next thing we are going to apply this pipeline, computational pipeline, to Down syndrome. As you can see, we start with a nonlinear dimensional reduction and clustering, and then we infer control and disease trajectories, and by aligning them together, we can obtain a disease-specific path. In this particular case, we identify C17, C18, C4, C5, C7 to C19 as one of the disease-specific developmental path. So this is how the pseudotime varies in this path. By applying pseudotemporally ordered gene expression data, we can infer causal link between transcription factors and target genes. This figure shows the top-ranked causal links. Then we compared the causal links with these causal links with a gene regulatory network obtained using CENIC. CENIC is a well-known gene regulatory network inference technique. You can see these solid lines provide the causal links that are supported by the GRN. So we use control and disease cell trajectories to identify disease-specific developmental paths, and we use convergent cross-mapping to infer causal links between transcription factors and target genes. As future work, we plan to compare it with ground truth gene regulatory networks and other causal inference techniques such as sincerities and scribe, which are based on Granger causality. With that, I would like to thank Dr. Wang, Dr. Anita for their support and guidance. Also, I would like to thank Dr. Wang's research group for their support. And finally, NIH, NSF, and UW Medicine, or University of Wisconsin Medicine, for their financial support. Thank you.